Hi, what we'll do today is compare and contrast living matter and inert matter. For reasons that are not totally clear to me, people take a lot of the amazing properties of living matter completely for granted. But if you think about them from an engineering perspective, they're absolutely remarkable. Each one of us started out as a single cell, and now we're dynamic composites of 37 trillion cells. You can cut off your fingertips and they'll regrow. Bacteria copy themselves precisely in 20 minutes or less. Our brains are incredibly powerful electrochemical computers that rival or exceed the performance of even the most advanced dry computers based on alternative architectures and materials like silicon. So there's nothing, of course, about this at all. It's just that we've grown up in a biological world, and all those remarkable capabilities of living matter seem completely obvious, but they're not. Imagine being able to harness all of those exceptional capabilities to do a lot of good and to do interesting things in health or climate or making things or providing more nutritious foods and so forth. Here's a Xenopus laevis, an example of living matter. It started out as a single cell that was fertilized. The single cell began to divide, and in the course of only a day or two or three, you end up with a much larger number of cells that are beginning to move and invaginate and gradually form the basic shape of the animal with a head, an eye, a backbone, and so forth. And after only a few weeks, you end up with a mature Xenopus laevis. If we compare and contrast living matter with inert matter, it can sometimes be difficult to figure out exactly what the differences are. If we think about living matter from an engineering perspective, we can ask questions about yield strength or elastic modulus or how much does it weigh. But all those comparisons would miss essential features of living matter. Let's compare and contrast two objects with four legs. Both of these objects are able to move stuff around. Let's look at a movie that came out of Boston Dynamics just a year and a half ago. That was remarkable. We saw a robot that was able to get up when pushed over. The robot was able to pick things up, to move around, and so forth. And on the face of it, those characteristics are quite similar to what a donkey is able to do. If we think about these two objects in terms of lifting capability or the number of legs or things like that, the differences are not particularly clear. But if we think about inert matter and living matter from the perspective of what you might call a life cycle analysis, the differences all of a sudden become completely obvious. How much energy is required to make a donkey versus a robot? How much does each one cost? Who owns the intellectual property? How do you fix it when something breaks? How do you feed it or power it? What are the waste products like? What's the environmental impact like? What's the fault tolerance like? How do you transmit blueprints? How do you change a robot versus engineering living matter? How do you make many more copies of each of those objects? Those are some of the questions that make it completely obvious what the advantages and limitations are of living matter versus other forms of matter. What we're going to do now is look at 5 or 6 more specific characteristics of living matter. Your brain is amazingly efficient. It dissipates approximately 100 watts, and every second there's 10 to the 16 synaptic events as you listen to this lecture or watch a movie or whatever else. If you compare these two numbers, 100 watts dissipated versus 10 to the 16 synaptic events, it's clear that your brain is incredibly energy efficient, because if you compare the joules per operation of the brain, 10 to the negative 15, an incredibly small number, to joules per operation for a computer chip that you have in your iPhone or in your laptop, there is a 6 order of magnitude difference. That's a difference of 1 million. So your brain on a per operation basis is 1,000,000 times more energy efficient than a lot of the other compute architectures that you may be familiar with. Unlike living matter or devices or objects fabricated from inert matter typically have a very small number of parts. Look at this steam engine. We're dealing with something like 100 parts, mostly steel, maybe some bronze and some other metals. And this machine generates mostly heat and a tiny little bit of work. So unlike something like the brain, you're dealing with an object here that is much less energy efficient and has an incredibly smaller number of different pieces. In living matter, you typically find a very large number of things working synchronously. For example, if you jump up, 1 trillion cells in your leg have to work together to, in aggregate, bring about the motion. And 1 quadrillion, 10 to the 15 motors have to coordinate their activities for you to be able to jump off the ground. As you think, as discussed, quadrillions of ion channels are flipping open and closed. A quadrillion is an incredibly large number, 10 to the 15, that dwarfs any kind of component counts or part counts that you might encounter in typical engineering. How difficult is it to make living matter compared to some of the other things we're familiar with? What do you need to make a tree? Light, air, nitrogen, carbon, water. These precursors are ubiquitous and they're cheap, and they're available anywhere on Earth that can sustain life. By contrast, if you think about the stuff you need to make a car, it's all across the periodic table, as well as leather, cotton, steel, quartz, other things you might mine, and also more complex molecules like plastics. Not only do you need a much larger number of different precursors to make something like a car, but you also have to move these materials across the globe, depending on where the material is found, where the material is produced. So you need a very complex logistics infrastructure to move this stuff to where the car is ultimately assembled. So a car is a global product that requires an incredibly complex infrastructure to bring everything together at the right place and then make the car. And that's completely unlike most situations in biology, where none of this is important and living matter is produced from materials that are easy to find in your immediate environment. Another feature of living matter is that it's intricately organized on multiple time and spatial scales. If you take a microscope and focus in on a car, it gets regular and boring incredibly quickly as you zoom through a big block of steel, for example. But what happens if you focus in on living matter with your microscope? You would start to see individual cells Those are the colored dots you see in this image, the small little colored dots that are red, green, or yellow, and so forth. If you keep zooming in with your microscope, it gets more interesting. You start to see individual neurons, and you start to see how those neurons are connected. And you can keep focusing in until you see molecules and ions and folded proteins and lipid bilayers and so forth. So in living matter, you're able to focus in over many orders of magnitude from nanometers to meters and keep discovering new things, new forms of spatial organizations, new kinds of patterns, unlike when you zoom through a car or a block of metal or a computer or something like that. Just those basic facts and features about living matter are already enough to transform a lot of different fields, whether it's how you make things or how you keep people healthy, how you fix the climate. But in addition to those characteristics, you have even more amazing properties, the amazing properties to self-organize or self-replicate, each component to reproduce the whole, to heal yourself, and so forth. What's really important to the latter category of capabilities are exponentials. If you take a single cell and it doubles, 2 doubling again, 4, 8, and so forth, due to exponential growth, you very quickly end up with an incredibly large number of copies of the initial object. And that is critical for properties such as development, distributing things across the globe, and so forth. There are certain tasks that sounds like complete science fiction if you think about them from a traditional engineering perspective, but are actually quite straightforward if you use living matter. For example, let's just say I told you, build me something that copies itself perfectly in 20 minutes. From a standard engineering perspective, you would say, how is this even possible? But in biology, it's completely straightforward. For example, if I tell you to build something that can spread itself across the globe very quickly, what if I asked you to change the world's climate for free? What if I asked you to figure out how to make some of the world's most complex chemicals with molecular fidelity? What if I asked you to store the Library of Congress on a pinhead? And all these things sound like complete science fiction, but are quote-unquote pedestrian capabilities of living matter. Let's look at bacterial cell division. Here's the movie. The single cell elongates, divides, elongates, divides, elongates, divides. Very soon, after only a few hours, you have thousands of copies of the original cell. If you're a starfish, you're able to regrow limbs. If you're a lizard, you're able to regrow your tail. If you're a planarian, someone can come and cut you into multiple pieces, and each one of those pieces will survive. It takes about 7 days. And after you've cut the planarian at day 0 into 3 pieces, after 7 days, you now have 3 small planarians. Of course, they're smaller, but each of those will live. Final point for today is to think about the common API for biology. If you look at lots of different things of living matter, like plants and people and elephants and microbes, you see an incredible diversity of form and function. But underneath all of this is a universal common blueprint, DNA. And what that means is if you understand the DNA code, and if you develop technologies for sharing those blueprints and editing those blueprints, and you can print more DNA, then all of a sudden you can make or engineer or tinker with all of biology. And that brings with it great promise, but also, once you think this through, it raises any number of significant concerns, some of which we will cover in our class. So the final thought for today is because there is a common mechanism for storing information in biology, you only need a relatively small number of technologies to broadly alter all of living matter, all of life. And those technologies are tools that allow you to design genomes. You could call this CAD life. You need technologies for printing the DNA once you've designed a genome. You then have to encapsulate the DNA. And then all you need is a trip to the grocery store to buy very simple precursors that allow you then to make a large number of copies of the object of the living matter that you've designed and built. I hope you've enjoyed yourself today, and I look forward to meeting you in class next week.